Okay, we're going over chapter 17. Chapter 17, 18, and 19, they're all cardiovascular related. So today we're just going to look at the part that is related to the blood. Okay, um, so let's go ahead and take a look at the overview of the blood circulation. As you know, uh, there is a reason for the blood to go to the different areas, and primary reason is oxygen. Oxygen needs to be delivered, and also the nutrients needs to be delivered. And usually what happens is there are different type of blood vessels. We will look at it later, but one of the type of blood vessel is called capillary. Capillary, they have very thin, is made out of simple squamous epithelial cells. So here is the wall of the capillary, right? So nutrients, oxygen, things in that nature, uh, they can pass through. All right, it should be in here. So let's just say oxygen, and then it reaches the cells. These supposed to be our cells or tissues, and they take up the oxygen and they give off their carbon dioxide as well. Carbon dioxide can go in now, nutrients, vitamins, glucose, things in that nature. That's a primary role of the blood. All right. So usually blood flows from the lung. All right. So from the lungs where blood oxygenation is occurring, you get rich in oxygen. And again, eventually what happens is the blood vessels will go back to the lungs to do the exhalation. All right. Along the way, obviously, they're going to have to stop by the heart. So the heart can, pump the oxygenated blood to the tissues, which we will discuss the role of the heart a little bit later. So let's go ahead and take a look at what is blood made out of. All right. So blood is the body only fluid tissues, and it's made out of formed elements. All right. And plasma. Fourth elements is erythrocytes, also known as RBCs, and also leukocytes. Leukocytes is for white blood cells. You might have heard about leukemia, right? This is erythrocytes. Leukemia is a cancer of white blood cells. That's what they are referring to. Okay. Now, there's also platelets. Platelets is for blood clotting. All right. There are certain proteins, factors, and factor VIII, and fibrinogen, and other proteins that are involved in blood clotting in case you have a rupture in the blood vessels. Plasma, so these are about 45%, and the plasma is about 55%. Together they make up the 100%. So, um, what is inside of the plasma? Well, the plasma actually consists of mostly water. That's what it is. But uh, plasma also contains protein. You might have heard about albumin, globulin, clotting proteins. So there are a lot of proteins, and a lot of these proteins actually being made uh, in the liver. All right. There are some stuff that is not protein really, some of waste acid. You might have heard about lactic acid, urea. All right. So usually those are the waste. All right. Keratin, but the urea is the most important one. And again, there are nutrients in the plasma. Some of which I can think of, it's like, amino acids, glucose, carbohydrates. Uh, then you have plenty of different ions and electrolytes, some of which are cations, anions, etc. And some of them sodium, potassium, calcium, uh, chloride, uh, bicarbonate. All right. And there are also gases, oxygen and carbon dioxide. All right. And the most and majority of it is actually water. So that's the plasma portion of it. Okay. And something, if we wanted to kind of like get an idea about this percentage, because the percentage might actually vary. Why is it going to vary? Well, first, let's go ahead and take a look and define what is hematocrit. Hemato refer to blood. Hematocrit is basically the percentage of red blood cells over total blood volume, which again, it's really about 44%. Okay, it varies. And in a minute, you'll find out why. Let's go ahead and take a look at this hematocrit. Okay, so a blood from an individual have been withdrawn, then placed into a centrifuge machine. Centrifuge machine uh, rotate this vial at the high RPM. All right, so that's centrifuge. All right, the outcome after a minute or so rotation, all the heavy stuff will go towards the bottom. All right, and the lighter stuff would stay on top. So the lighter stuff is staying on top is the plasma. So here's the 55%. All right. Usually, the leukocytes and the platelets, they're known as a buffy coat, happen to be right in between, and the erythrocytes happen to be towards the bottom, which we mentioned is a formed element. So, how do we figure out the hematocrit? And what does the hematocrit tell us? Well, tells you erythrocytes in this case is a 45%, more or less. So, one way to determine the hematocrit is, let's go ahead and take a look. Let's say that you had this vial, okay, and let's say half of these was a red blood cell and half of it was plasma. All right. So you could literally measure from here to here. All right. say is 1 centimeter. Okay, that's the erythrocytes. And from here to here is the volume, right? So that would be, let's say, 2 centimeter, right? Percentage of the red blood cells. In this case, that would be 1 centimeter, right? Over total blood volume. In this case, would be 2 centimeter. And now you could see is multiplied by, percentage. So you need to multiply it by 100, and that would be 50%. In this case, hematocrit happened to be 50% because it's half and half. Now, what if a person undergoes dehydration? In other words, think about what happens when you are dehydrated. Okay? What will happen when you're dehydrated? Right? Obviously, you're going to lose water. That is what's going to happen. Okay? So if you lose water, earlier we mentioned uh, water was in part of the plasma. So if you wanted to correctly draw, for instance, a person that is losing the water, obviously, let's say it's the same person. After running a marathon and not drinking water through sweat, you lose a lot of water. So now here's your red blood cells. You didn't lose any blood, but you lost water. So your water amount is going to be lower. All right. Now, from here to here, it still would be 1 centimeter. All right. But the total volume no longer is 2 centimeter, is less than that. Let's say it's a 1.5 centimeter. Okay. So as a practice, you could go ahead and see what will happen to the hematocrit. Does the hematocrit increases? Does the hematocrit decreases? All right. In order to answer that, you need to take a look at what is happening and what is hematocrit. Percentage of the red blood cells. To me, the percentage increased in this example. Take a look at it. No longer they have 45% or 50% in our case. Uh, they have more. Take a look. This is more percentage. If the whole thing is 100%, this would be, more like, you got to put it in the calculator to figure it out, but looks like it's way above 50%. All right. Now, another scenario. What if this patient went into hospital, same patient, because it's dehydrated, you know, they went to the hospital, he was unconscious. As you know, when the person is dehydrated, your blood pressure is going to be lower now, all right? Because the volume of the blood no longer is the same, it's dropping, all right? So your blood pressure is going to be lower. This is also the case when you donate the blood. But right now, we're not donating blood, right? We lose the water. The patient lost the water, went to the marathon, pushed themselves to the limit, didn't get hydrated before or after, went through this series of jogging or running and losing, and it was a hot day, and hours and hours of running caused the blood loss. I mean, the blood pressure to go lower. All right. Now went to the hospital because he was unconscious. Sometimes when you don't have enough pressure, the blood doesn't reach the brain, and you can faint. And right away, the IV was placed on the patient and was placed into the hospital bed, and the person is unconscious. But let's just say the nurse forgot to take out the IV, and the IV continuously, or another nurse came in and replaced this IV for the new one. So what is happening now? As the time passes by, obviously there's going to be more fluid in the person's body, all right? So if there's more fluid, remember, you're putting like, isotonic solution to the person. okay, to raise the blood pressure. But if you do it too much, in our example, or a person gets way too hydrated, now scenario is going to be different. This is after IV, too much IV, all right? Well, let me go ahead and ask you, can you guys think about or draw? You don't have to be exact, just draw the possibility of the hematocrit. How would you draw that? Knowing that you have a lot of IV, that means a lot of electrolytes and water. What does that mean? That means increase in plasma. All right? Pause the video and see if you can draw hypothetical hematocrit with lots of plasma. All right, so the blood is saying you didn't lose any blood, but now the IV, because of putting your IV, this is more. All right, so you can see the amount. This is way more now. Again, if you wanted to do the hematocrit, uh, let's say from here to here is 1 centimeter. All right, now the total volume is more. Okay, so 2 centimeter would be up here. Maybe we could put like maybe 2.5 centimeter. So again, it'd be 1 over 2.5 multiplied by 100 to get the percentage. And again, you could calculate it to see what happens to the hematocrit. Does the hematocrit go up? Does the hematocrit go down? And what are the reasons for it? Again, take a look. You'll see this hematocrit is lower now, all right? Because there's more percentage of the plasma. So I hope that those examples helped you in understanding of it. I hate to see individuals, just memorizing a bunch of terms. Okay, it must be 55, and not knowing even what they're doing. And also on top of that, you never know. Maybe I'll use a different scenario that you need to calculate really quick and so on. Uh, the problem with online classes is people, they always wanted to look things up without thinking about it. And unfortunately, looking things up might help you a little bit in terms of grading, but in terms of understanding of it is not going to do much. All right, let's go ahead and take a look at the characteristics of the blood. All right, so now that we learned the hematocrit, let's go ahead and take a look at some of the characteristics of the blood. Blood is sticky and opaque. In other words, it's not transparent. There's all kinds of fluid, there's all kinds of protein, there's all kinds of electrolytes inside of the blood, so it doesn't make it transparent. Color may vary, really depends whether we're looking at oxygenated blood or deoxygenated blood. Right? Again, color varies. Oxygen poor blood, usually they have a darker red, and you've seen like maybe on TV on different shows that the person is losing blood or died recently from you know losing blood, got a gunshot for example, and you notice like the color changes to a little bit like a darker, sometimes even bluish color. All right. Blood pH is about 7.35 to 7.4. This is an average about 7.4. We say, make it simple. 8% of the body weight. Average of the blood is about 5 to 6 liter in male and 4 to 5 liter in female. Now, keep in mind, the females usually they are a smaller individual compared to the men. Also, uh, female undergo menstrual cycle and they're losing a little bit blood there. So again, those explains why slightly there's lower amount of volume in female blood. Okay. All right, let's go ahead and take a look at the function of the blood. Real quickly, there are three functions basically. You have substance distribution, you have regulation of the blood level of substances, and you have body protection. All right. So the first thing is I would like to take a look at is distribution. Can you guess what are you trying to distribute? We mentioned it at the beginning a little bit. All right. So what is blood is trying to distribute? What is inside of the blood? We mentioned this already. So that might help you. You could pause the video to see if you can name and think about all the things uh, that the blood is distributing. All right, one, I'll give you a minute to think about it. All right. If you mention gases, good job. Oxygen and carbon dioxide, they need to be transported. Nutrients, they need to be transported. All right. So those two we already said. All right. So if you're able to pick those up, that's fine. Waste, you know, blood needs to get filtered in kidneys. Kidneys get rid of the urea and the waste, nitrogenous material in our blood needs to be transported and exited out. All right. So the urinary system and the kidneys in particular filter the blood in a continuous basis. All right. People with kidney failure, they need to go to dialysis center. So their blood goes into the machine, it gets filtered and returned back into the body. All right. So hormones, hormones also can get distributed. All right. Uh, Endocrine system uses blood to transport many of the hormones to the target cells. So again, we can see a lot of substance distribution taking place. All right. Sometimes this might help you. Uh, Blood vessels is kind of like a freeway, and the cars, people going back and forth and so on, those are kind of like blood. They need to get to their destination. They use the blood vessels in order to get to the destination. So you get to your car, you go to the freeway, you get home. And the home is kind of like analogous to the tissues. All right. I don't know if these helps you or not, but you could kind of like relate things to the daily activities. Sometimes it might help. All right, next up we go to the regulation of particular substances. What are we trying to regulate? Well, temperature is one, trying to regulate the temperature. So when you're cold, for example, you have a tendency to keep the blood towards the center. That's why your hands feel cold, your feet may feel cold. All right, and vice versa. When you are exercising, the blood flows to the muscles. We discussed the sympathetic activity already, so because they need more oxygenation and so on. So um, um, regulating the temperature and so on is important. All right. Uh, they can absorb and distribute the heat as your muscle contract. They generate heat, and this heat needs to be dissipated, needs to get distributed throughout the body, so that way you could get rid of it. Otherwise, it's as if though you were getting a fever, and when you have a fever, your body temperature goes up, and your enzymes are not going to be working. Remember, factors that were affecting the enzyme. One of them was temperature. This is the reason why when you get a fever, you can't eat, can't do exercise, can't do much. You have to stay home and rest. And hopefully, it's not a coronavirus. Okay. So another thing that you are trying to regulate is actually pH. pH, potential hydrogen or acidity and basicity. And we have a buffering system, bicarbonate that we use. But to be able to monitor that, again, one of the primary organs that does this actually is kidneys. So extra acid can be getting rid of through your urine. Also, there are buffer mechanisms that can neutralize the acid and bases. As you know, if you have too much acid in the blood, your blood is going to become acidified. This is called acidosis. And alkalosis refers when your blood is alkaline, is basic. You don't want neither. Again, another factor that affected the enzyme was the pH. If you cannot monitor the pH, remember it was around 7.4, you need to regulate that. If it gets too acidic, now a lot of enzymes they lose their function. Right? If it gets too basic, same problem. In fact, actually more problem occurs if your blood is too basic than too acidic. All right. Uh, Alkalosis can cause number of problems, but the body is really the buffer is very limited when it comes to alkalosis. Anyways, that's what we need to regulate. The fluid volume uh, also needs to get regulated. As you know, in a previous example of a person being dehydrated, the volume went down. That's a big problem because now there is not enough pressure to send in the oxygen and carbon dioxide to a proper area. And one of the examples that I told you is, uh, you know, individual that lost too much water. Sometimes it faints because of low volume. If you have a low volume, how are you going to get the oxygen to the brain? And if it doesn't get that, it just faints. All right. So you need to administer or have the person drinking water at that time. Too much volume is also problematic. People sometimes, they drink water, but for whatever reason, maybe because they're traveling, maybe they are on the road. A lot of truck drivers, by the way, you know, they can't find the restroom and they hold it in and they keep drinking because it's hot summer days. All right. So in this kind of thing, what happens is the volume goes up. There are other things that could cause the volume to go up. As you know, salt can increase the blood pressure because the volume goes up. And actually, the idea of it, you've seen it before. So as glucose. So when you have way too much solute, whether it's a glucose, whether it's a salt, your tissue loses water. Go back to the experiment of the osmosis that we discussed. So water is losing from your cells. So your cells is become dehydrated, you're losing the water. But because the solute attracts the water, your volume is going to go up. So your blood pressure is going to go up. The reason why people with glucose, too much glucose, hyperglycemic or diabetic, drink a lot of water and they go to the restroom more often is exactly the same reason. They get dehydrated, so they're always thirsty. And because you have too much glucose as a solute now, all right, your blood pressure is going to go up now. These are all big problems, all right? And they're always thirsty. So they're drinking water to restore the water that they lost because, keep urinating and so on, all right? So those are the important in particular substance uh, regulation, all right? Uh, Next up, we're going to go ahead and take a look at protection. Protection mostly is the job of the white blood cells. White blood cells, and we will take a look at the category of white blood cells, but usually they defend against the foreign invaders, pathogens, diseases, bacteria, viruses, things in that nature. And there are certain type of white blood cells that they produce antibodies, plasma cells, and uh, lymphocytes, in particular B cells. All right, B cells comes from the bone marrow. That's why it's called B cells, and and they do not need to get matured in the thymus, but they produce antibodies. One of the experiments that I kind of like selected for you is related to the antibodies. So hopefully uh, we can get to this part a little bit later. But there are protein molecules to attach to the antigen and attack something that is not recognized by immune system. These are called immune response. All right. Interestingly enough, one of the methods to treat coronavirus is happen to be taking the antibodies from the person that recovered. In their blood they have antibodies, they have these proteins that probably recognize and already combated the virus, and they try to give it to the individuals that are having a immune compromise or perhaps weak immune system, and they try to have this blood. Now they have the antibodies or protein molecules to recognize the foreign protein from the COVID-19, for example. Okay, that is the idea. Uh, Next up, I would like to go over a few other things uh, and talk a little bit more about the formed elements. All right, so you could rest the rest, you could read the rest. So the formed elements we mentioned, platelets, erythrocytes, and leukocytes. The white blood cells are complete cells. What that means is they have nucleus, they have all those organelles, etc. Red blood cells is not so. When you're looking at the mature red blood cells, they actually lost their organelle, they lost their nucleus, and platelets are just fragmented cells using for blood clotting. So most formed elements, they can be in the blood stream, but they're not going to be able to live forever. All right, so they need to be renewed. Red blood cell life expectancy is about 100 to 120 days. So now you have to renew them from the bone marrow. All right, if you recall your anatomy, um, here is the bone, inside was the bone marrow. This is where they get renewed. All right. So it depends on your needs. If you have an infection, hey, we need more white blood cells. White blood cells get more manufactured. If you need more red blood cells get more manufactured. You need to replace one, they replace it for you. So it really depends on the bone marrow. Let's go ahead and take a look at shape of the red blood cells. The red blood cells, as you know, they are biconcave. Look at the concavity, one side and two side right over here. So this is a top view. They did like a cut right in the middle, and this is from the side view. You're looking at it. So they are biconcave. All right, they don't have any nucleus, no organelle whatsoever, but they do have a lot of hemoglobin. All right, hemoglobin is a protein molecule that is important in transfer of gases, that is oxygen and carbon dioxide. Um, Red blood cells plasma contain a protein known as a spectrin. What is the spectrin? There are other proteins, by the way, as well. Hemoglobin was another one. A spectrin give the red blood cells flexibility. All right, and allows them to change their shape only if they need it. Let me give you an example of that. Here you have a capillary. All right, let's say before you get to the capillary, you have arterioles. Here's arteriole. Here is capillary. All right, so red blood cells are large. As they come into the capillary, they need to change shape now. See, they are changing shape as they're passing through. This would slow them down, obviously, but it allows them to go one by one. In the capillary, only one red blood cell at a time can pass through. And because they come into a contact of the wall and they are going very slowly, that allows them to exchange the oxygen to the tissues, for example. Right? These are the cells of the tissues, for example. So oxygen can go in here, and again, carbon dioxide can come out and being carrying away. Here's a carbon dioxide, or some of them in which it could go to the plasma as well. We'll discuss what percentage of it could go where later. Okay, but the idea is that a spectrum allows it to change shape, and that's pretty important. That gives it the flexibility. What it reminds me of uh, is actually, I don't know if you ever had this, but there are boba drinks. All right. Uh, there are, some tapioca balls like right here. It looks like that, and then you have this straw. As you're taking up the drink, which is similar to the plasma, you're also taking these, boba, but only one at a time passing through. But the boba are very flexible, so it could go right through your straw. Your straw is usually larger than the normal straw, and tapioca's is just very flexible, so they could go in. And that gives you more time to finish up your drink or taste it one at a time, etc. And it's very popular, as you know. The point is, one of these at a time passing through, similar to what we discussed. All right. Now let's go back to take a look at here. Now, this biconcavity, it allows more surface area. So red blood cells, compared to the volume that they're holding, they have huge surface areas. So again, more chance of exchanging of gases back and forth, okay, because they have a lot of these surface area around, all right, compared to the volume that they're holding. If it was a spherical, you would have less surface area compared to the volume that they're holding. The volume is going to be a lot more, obviously. So if you take away the water, all right, 97% or the majority of the red blood cells are hemoglobin. And hemoglobin is quite important, as you know. Now, ATP is being generated anaerobically. As you know, ATP is necessary for the energy, but if they use the oxygen that they are carrying, so here is the hemoglobin, for example, just giving you a huge hemoglobin so you could see it. And here is the oxygen that they carry, and you have millions and millions of these hemoglobin in a red blood cells. But if you consume this oxygen to generate ATP, what will happen if it doesn't happen? But if they generate the ATP aerobically, then you literally will use up a lot of oxygen that are supposed to be carried and shared with the tissues. So by the time they get to the tissues, they don't have anything to offer them. All right, so the tissues would be demanding the oxygen, and they're not going to be working, obviously. All right, if you don't get the oxygen to the brain, you get a stroke. If you get no oxygen to the heart, you get heart attack. You get no oxygen to the kidneys, kidney failure is occurring. So across the board, you'll see that um, you will have a lot of problems. So I give this analogy. If you have a restaurant or you're in offering food, if you consume all the food, you wouldn't have anything to offer the customer. Customer comes in and want a particular food, you're running out because you already ate it or you give it to the family members or didn't have it. You used it already. All right. As you know, soon you'll be out of business. All right. Um, so we can kind of like go in to the hemoglobin at this point in time and talk a little bit about that as well. Here's the molecule of hemoglobin. It consists of four chains, two alpha and two beta. And you can see the name of it right over here. That's not that crucial, but it has a protein which is made out of amino acids, and they are in a quaternary structure because it's more than two subunits, it's four subunits. Each of these subunits have a hem group. This part right over here is called a hem group, all right? And inside of the hem group, if we go ahead and take a look at it, this is a hem group, all right? You see Fe. Fe is for the iron, okay? So in the metal, they have the iron in it. So one of the major ingredients for the hemoglobin is having iron. As you know, meat has iron, spinach has iron, a lot of food have, seaweed has iron, all right, in it. Okay, so if you want to make a pancake, you need certain ingredients. You need sugar, you need flour, you need egg, you need butter, things like that. If you do not have one of those ingredients, your pancake is not going to come out right. All right. Same thing in here. You need amino acids. Amino acids because hemoglobin is a protein. So to put up this chain together in a way that they are and so on, you need a lot of these amino acids that are bonded together by different type of bonds, hydrogen bonds, covalent bonds, interactions, and so on. All right. So that is important to think about. So protein globin has two alpha and two beta and one hem group, as you know, and each hem group can actually bind to one oxygen molecule. Oxygen molecule is O2. So you could put in one O2 here, one O2 there, one O2 here, and one O2 there. So a particular hemoglobin can actually transport up to 4 molecules of oxygen. And that's just one hemoglobin. Remember, in the red blood cells, you have so much hemoglobin. All right. So another important issue is having a right amount or concentration of the hemoglobin. If your red blood cell is normal count, like you counting it, all right, and it's normal, but you don't have that much hemoglobin, you're still not going to be able to deliver the oxygen. Hemoglobin is kind of like a taxi cab or Uber driver, for example. Each Uber driver can have 4 passengers, all right? So that's the limit. You go from one area to another, all right? Drop off those passengers and pick up a new passengers, carbon dioxide, for example, all right? So when the oxygen or when the hemoglobin taking the oxygen to the tissues, they will unload them and they will load up carbon dioxide, all right? And these are exchange of the gases, as you know, all right? So this hemoglobin, they have affinity for oxygen. It's not exactly the same, but think about it as a magnet and a metal. Right? They have affinity. When you come into a close or you have a lot of magnet or you have a lot of metal next to the magnet, they stick to one another, right? So it's kind of like that, all right? And hemoglobin that are attached, they have different terms now. Carrying the oxygen, they call it oxyhemoglobin. This is when the hemoglobin bound to oxygen. If they unload the oxygen, then it will call deoxyhemoglobin. And when they attract and unload, they load the carbon dioxide, then it's the term called carbaminohemoglobin. That means, hemoglobin bound to the carbon dioxide. Okay. Uh, let's go ahead and briefly talk a little bit about the process of hematopoiesis, which is a blood cell formation. Again, it happens in the bone marrow of axial, skeletal, and pelvic girdles. Your thick bones, they have bone marrow in it. Uh, Majority of long bones, humerus, femur, they're capable of doing that. And as a result of that, you can manufacture more red blood cells if you need it, or perhaps if the red blood cells have been damaged and they need to be replaced, or they just got on too old. All right, both of which they need to be made. So hemocytoblast give rise to all the form elements. Again, think about the form elements, white blood cells, and all that too. All right, so let's go ahead and take a look at how we can actually make erythrocytes. There are different phases, so you could kind of like take a look at this to show it to you. So this is the stem cells. This is hemocytoblast. This is where kind of like everything starts off with. Something about the stem cells. Uh, Stem cells, they have a capability to become a different type of cells. All right. Nowadays, when a person is giving birth, they ask the parents if they wanted to save the umbilical cord because the umbilical cord contains the stem cells. And you could bank that for some fees or whatnot. The idea is when the technology comes, maybe you could use these stem cells for a purpose that the person might need. For example, a person accidentally fell off of the horse after grown up, and now they need the nerve. Now, where you're going to get that? The stem cells might be the key uh, for this person to walk. Again, the technology is not there, but the stem cells have there, and some scientists actually they disregard the ethnic, they disregard uh, the idea of what can they experiment on. So some of the babies that are born prematurely and so on, they look at the stem cells and so on. So in terms of the ethical standpoint, you know, you're not supposed to be doing uh, any type of research on, you know, uh, humans, but I'm not talking about clinical trial. Anyways, committed cells, they become committed cells or pro-erythroblast, and then they become erythroblast. All right, pro means before. Okay, so they become destined to become the erythrocytes. Take a look at what you start off with. Take a look at what you end up with. A lot smaller, right? No nucleus whatsoever, right? So let's go through these stages. Stage one, ribosome synthesis happens in an early erythroblast. In other words, as you know, ribosomes are being made in the nucleolus, and ribosome was a site of protein synthesis. So you're trying to set the stage for protein synthesis. Next up, we get the hemoglobin accumulation in the late erythroblast. So right now, we're looking at the erythroblast. At the later instance, what happens is now you're utilizing the ribosomes and you're making a lot of different protein. In this case, hemoglobin. All right, and this continues the same thing. This is the later stage, and then the phase three is ejection of the nucleus. All right, from normal blast, ejection of the nucleus taking place. In other words, what happens is now you're looking at reticulocytes without any nucleus. Nucleus got ejected for a reason. One, you have less weight and more flexibility. All right. If you are traveling somewhere and you have extra luggage is kind of similar to uh, nucleus. All right. It would be hard to travel. It would take you longer. Speaking of travel, as you know, nobody's traveling these days. But that's what happens. If you have just a carry-on or just by yourself, you can get to your destination a lot quicker. All right, and you have more flexibility. Same thing in here. Uh, Not having this nucleus, it gives the red blood cell more flexibility, less weight, so they can get to their destination quicker and deliver what they needed to deliver that we discussed already. All right. So the regulation of making red blood cells is obviously important. If you have too many red blood cells, what happens is your blood cell become more viscous. All right, viscosity increases when your blood is thicker. That causing problem now. Why is it causing problem? You might say, well, I have too many red blood cells. That's a great thing. A person can get a lot of oxygen. That's not true. All right. The reason is when you're looking at way too many, all right, they could actually come into the contact of one another and come into a contact of the wall of the blood vessels. As they come into a contact of wall, they slow down. All right. And now you need increasing blood pressure, all right, to deliver the same amount of oxygen. Imagine that there are many cars in the freeway, all right. You're going to get into traffic jam, all right, because there are more friction. There's more coming to contact of one another. You can't speed up anymore, all right. So it adds to the congestion. It's the same idea, all right. Um, we can go ahead and take a look at this scenario. If you have two syringes, similar size, one of the syringes is filled with just water, let's say 50 cc. The other one, let's say you filled it up with honey, 50 cc. So this is honey, this is water. No? Okay, now if you want to uh, eh, sure, withdraw the same amount at the same time, which one of these you have to add more pressure? Obviously, the honey. Honey is viscous. So when you pour it out, it takes time, all right, to come out. All right. So you have to put in more pressure in here, right? And blood pressure is going to get increased to get the same amount out rather than water. You don't need to put too much pressure in there. All right. That is for the given time. That's the idea. Um, about too many red blood cells. Another problem is when you have too few. If you have too few of the red blood cell, you might develop what's known as a hypoxia. Hypoxia is a condition when the tissue is lacking oxygen now. All right, so that's problematic because again, the tissues won't do very well when they don't have the oxygen. Remember, every tissues in your body requires oxygen. You got muscles that in order for them to contract, they need oxygen. People with hypoxia, they're always sleepy. They always wanted to rest. They get exhausted if they get up and they do a little bit of exercise. So they don't have enough red blood cells. They're always cold, temperature regulation. All right. Their hands and feet always so cold. All right. They don't have much of the energy throughout the day either. All right. So again, these giving you an idea about what happens. So there's got to be a balance. How many red blood cells you have or produce versus how many red blood cells are dying. The amount of red blood cells should not fluctuate that much. It's not supposed to go up. It's not supposed to go down. If the rate of destruction is less than rate of production, all right, that means red blood cells is going to go down. And vice versa, if the rate of production is more than the rate of the destruction, then, it will be more viscous. You need to have that balance, all right? So erythropoiesis are controlled hormonally, all right? And again, it also depends on the ingredients. Some of these ingredients I mentioned, vitamin B, amino acids, iron. Again, think about the pancake that requires right ingredients to be made. All right. So let's go ahead and concentrate on this hormone. Where is this hormone coming from and what does it do? It comes from the liver, but majority of it comes from the kidneys. So I want you to remember kidneys. Erythropoietin or EPO released by the kidney. So it's a hormone, all right? So uh, tissues of the kidney, once they experience hypoxia, they start generating this EPO. EPO will get into the blood because hormones, again, substance distribution, all right? That was another function of the blood. They can get to the target cells, all right? So due to lower amount of RBCs, you get hypoxia. Now you have low oxygen availability, and the demand for oxygen is going to be increased. This, in turn, is going to cause an alarm, kind of like triggering more production in the red blood cells. All right. So EPO is going to reach the uh, bone marrow, and the bone marrow is going to manufacture more red blood cells. Here is the idea. Uh, so the first thing is going to happen is hypoxia, all right, lower availability of the oxygen due to lower of the red blood cells or other reasons, but all of these scenario, the oxygen level is going down, all right. Next, what happens is kidney and to the smaller degree liver releases erythropoietin. Remember, for our case, just remember it as a kidney, EPO, that's the abbreviation for the erythropoietin. And now what happens is erythropoietin stimulates the red bone marrow, and as a result of that, the erythropoiesis increases the red blood cell count, and you get back to normal. All right. Now you have ability to increase your oxygen carrying capacity. All right. So ability to carry more oxygen. All right. So a lot of factors could determine your load of oxygen carrying capacity. One of those would be the number of the red blood cells. Another one is the hemoglobin. Do you have right amount of hemoglobin? All right. Do you have a right amount of pressure? Do you have the right amount of red blood cells? As you can see, a lot of factors play important role. All right. One is the numbers. But what if the numbers is okay, but they do not have that much of a hemoglobin? So hemoglobin concentration should be measured as well to determine that. All right. Um, so the process of erythropoiesis require ingredients: protein, lipids, carbohydrates, iron, vitamin B2, B12, folic acid. All of those are important, obviously. All right. So body can store some of this iron in the hemoglobin, 65%, but also the liver, the spleen, and the bone marrow as well. All right. So there is a little bit of recycling going on. All right, so 100 to 120 days is the life expectancy for erythrocytes. The erythrocytes, they become older, they become fragile, and they need to be taken care of. They need to be remade, to be replaced, and these are what happens to them as they become old and fragile. Well, they can be engulfed by the macrophages. Macrophages are one of those phagocytic cells, phagocytosis, cell eating. So they kind of like they can chop it down and ingest it, kind of. All right. Some of these will get filtered in the spleen. All right. So the hem and the globin part gets separated, but iron can be salvaged for reuse. When you wanted to remodel your bathroom, you don't want it to knock it down and demolish everything. All right, certain things can be salvaged or reused. Your sink might have been perfectly fine. Your light fixture may be perfectly fine. Maybe you updated your tile or other things, but you didn't knock out everything. All right, because that would be costly. It would take more time, and it would be more headache. All right.
So more work is required. Same thing in here. If you wanted to make everything from scratch, that would be more difficult and more tasks rather than recycling some of the parts for keep reusing it. All right. So the hem group, all right, it can be degraded to a yellow pigment known as bilirubin. All right. So a liver secretes this bilirubin into the intestine in a form of a bile. Later on, I will discuss about what is this bile do. Basically, it helps to break down the lipids. All right. So the intestine uh, metabolize it to the urobilinogen, and then this pigment leaves the body in fecal material, and that's what happens. Now, what we wanted to take a look at what happened to the globin and what happened to the rest of the story. So the globin is metabolized and break down into amino acids. As you know, amino acids can be absorbed into the circulation. It could go different places. Our cell can use these amino acids for making their own protein. All right, so whatever that is, our enzymes made out of protein, antibodies made out of protein. Lots of things are made out of protein. So obviously, in order to make protein, even the spectrin, even the globin, even the albumin, they're all made out of protein. So you need ingredients, you need those Lego pieces, and these are the Lego pieces to put in together. All right, so uh, the HB look, this is kind of like the idea of what is happening. Uh, so on the first part, the kidney producing the EPO. EPO will go to the bone marrow making the red blood cells. But again, as they age and they're getting damaged, they engulf, the red blood cells engulf by the macrophages of liver, the spleen, bone marrow, and the hemoglobin is going to break down to two parts. Here's the hemoglobin. Let me increase it so you can see it a little bit better. All right, one part of it is the globin, which is the protein part. So that would be metabolized into the amino acids and it is released into the circulation. From then on, it could be utilized by our cells. What about the hem group? If you remember, the hem group had the iron as well. So the iron can get stored into the liver and the rest of it basically becomes the bilirubin, which again, it can be converted to the bile and send into the small intestine, and the small intestine can utilize it to break down the fat and things in that nature. All right. So that was the idea or the life cycle in a nutshell. But what we wanted to do is take a look at some of the disorders, blood disorders as well. All right, so these are mostly red blood cell disorders. We will discuss, maybe some of the white blood cells and the blood typing on the next lecture, perhaps. But anemia is abnormally low oxygen carrying capacity. It's more like a symptom than the disease itself. The blood oxygen level cannot support the normal metabolic activity. Again, a person is fatigued, paleness, shortness of breath, chill, get always cold, low energy, sleepy, you need to rest, all of that because you don't have that much oxygen to sending into the tissue. Why is it so? Well, there are different type of anemia, as you know. One of these is called hemorrhagic anemia. Hemorrhagic anemia occurs acute, means right away, or chronic, it takes time, loss of blood. So you have a hemorrhage somewhere, you have internal bleeding somewhere, and you're losing blood, obviously. Of course, you're not going to have as many red blood cells now because you're losing them through your leakage. Hemolytic anemia is the next one. When the erythrocytes, before they become mature, they all of a sudden they lyse, they breaks, they rupture. Lytic means break. Hemo, blood. Aplastic anemia. This is a problem with bone marrow. destruction or inhibition of the bone marrow cause aplastic anemia, where there's a problem in the factory. If you have a factory but you're not getting your supply chain is interrupted. Now you're not going to get the item to sell. This is the same idea. Next up, you have iron deficiency anemia. This is the most common type. Many people have this problem. All right, two reasons. One, perhaps maybe there is not enough iron in the food, so your diet is poor. Okay, uh, it's a secondary result of hemorrhagic anemia as well, but also sometimes it's because you're not absorbing iron. In order to be able to absorb iron, you need maybe other vitamins in order to do that, and that is the reason why. So, uh, you know, it really depends. But again, having food that are rich in iron, meat and dark green vegetables, uh, you know, uh, spinach, seaweeds, things like that has a lot of good iron source, but it's still you need to be able to absorb them. All right. So deficiency in vitamin B12, that's another problem. Pernicious anemia is the result of that. We're moving forward to thalassemia and sickle cell anemia, which are the genetic problems now. Genetically, the DNA is faulty, and you're making a different amino acids, and as a result of that, you're going to have problems. Now, in terms of the sickle cell, you see a lot of, red blood cells that do not look normal. Normally, your red blood cells are supposed to be nice and round. These sickle cells, they're kind of like this. They're kind of like this. So you see a sign of sickle cell. Again, it's a genetic disorder. It's an autosomal recessive, as you probably looked at the genetic before. Uh, all of these causing a similar problem with each, which is low oxygen availability. This sickle cell anemia could be a serious problem because it could clog up the arteries, and again, it's not efficient for them to passing through, so it's also a problem too. Um, Moving right along, polycythemia, when the blood is getting way too viscous. We mentioned this already. For whatever reason, your blood is getting viscous. That's polycythemia. Now, there are other disorders that I didn't get a chance to elaborate, but interestingly enough, uh, you know, there are some pathogens that they can, occupy parasites, pathogens like malaria and things in that nature. That could be another blood disorder. Uh, People that excessively bleed, and they lack blood clotting factor. This goes back to the platelets, for example. One of those is hemophilia. Hemophilia, again, is a blood disorder where you're lacking the clotting factors, and platelets, they do not clot. So if you're shaving and you're bleeding for longer period of time, and you keep on bleeding, your platelets is probably low, all right? And or you may have those protein factors that are involved in coagulating the platelets together to, again, we discussed this in the positive feedback early in the semester, if you recall. But regardless, those are all problems, and that summarizes uh, the idea of uh, red blood cells. And I hope you guys enjoyed the lecture. We'll come back and talk about the blood typing and the system of white blood cells. I hope you learned something and enjoyed this lecture.